to introduce Dr. Christopher Ellis, our speaker tonight. He will be telling you about the Scottish rainforests. Dr. Ellis is an ecologist. He did his PhD at the University of Manchester, and he studied the paleoecology of Scottish peatlands. He worked at the University of Laval in Quebec, and then the University of Nottingham, where he was introduced to lichens. He started work at the RBGE in 2003, where he got to grips with lichens by training with Brian Coppins. As he was worked at RBGE, his interest in lichen diversity and how the diversity is patterned in the landscape, how it might be affected by global change, and what we might do to protect these species. So I'll now hand over to Dr. Ellis and look forward to hearing his talk. Thank you.
Great, thank you very much. Just, can you hear me okay, Jill? Is that working all right?
Yes, that's fine, thank you.
Thank you. So thank you very much for the invitation to give a presentation this evening. It is a real honour to have been invited to talk to the Botanical Society of Scotland. And I was asked to talk about Scotland's rainforests. So I will advance this slide. So I'm going to talk about temperate rainforest in Scotland. I'm going to introduce temperate rainforests in terms of their global importance and biogeography. We can't talk about temperate rainforest without thinking about lichens. So the second part of the talk, I'm going to introduce lichens, think about something of their biology and what makes them tick. And then I'm going to finish with 3 pieces of research that we've done quite recently that point to how we might better protect our rainforest, the rainforest that we have in Scotland. So in thinking about our temperate rainforest in Scotland, I thought I'd take a step back and as botanists, think about the way in which we organize our understanding of botanical diversity on the planet. The way in which we recognize that different kinds of plants grow in different places, in different habitats, in different ecosystems, in different biomes. And of course, that's these patterns of botanical diversity occur in relation to things like the availability of moisture or the distribution of heat across planet Earth. And so here's a global map that shows soil water. So this is the amount of soil water in relation to things like precipitation, for example, and evaporation and evapotranspiration driven by heat. And we can see that soil water isn't evenly distributed across the land surface. And we have areas in blue with very, very high soil water, and then areas in orange that are very arid with very low soil water. And as botanists, of course, we recognize that different kinds of plants grow in these different places. They're adapted to different water regimes. So we have a lot of soil water in a place like Scotland, particularly in northwest Scotland. Then we have our wonderful peatlands dominated by sphagnum, which is adapted to these very, very wet environments. But in places where we have low soil water, then we have plants with a different set of adaptations. So succulents and cacti and so on, like these in Arizona. And we can recognize the way in which plant species sort themselves according to these physical factors, the way in which they're adapted to these different environments, and they occur in different places with respect to those adaptations. And this has been recognized for a really, really long time. So here's a beautiful map, a 19th century map based on the work by the biogeographer von Humboldt. And it recognizes different biomes, different zones across the Earth, latitudinal zones. Some really nice ones here. We've got an equatorial or a torrid region, through a hot region, a temperate region, a cold region, to a wintry region here, and then a frozen region up in the north. And von Humboldt also recognized that these latitudinal gradients recapitulate themselves altitudinally. So in these beautiful diagrams here, we see different vegetation zones as you climb up an altitudinal range on a mountain, and that those zones also have, they are structured by latitude as well. So the effect of latitude and altitude that controls the distribution of plant species, and plants are adapted to these different environments. Now, if we leap forward a little bit, this kind of work has undergone lots and lots of revisions and modernization, people thinking about how we can divide the planet up into different ecosystems or different biomes. And this shows Holdridges life zones. So this is a bit more empirical. So this diagram is showing annual precipitation along this axis here and evapotranspiration along this axis. And those two factors create different humidity provinces. And cutting across this diagram, we also have temperature. So if we have very low precipitation and we have very high evapotranspiration, then we arrive at the desert biome. If we have very high precipitation and very low evapotranspiration, then we arrive at a rainforest biome. And into a scheme like this, we can position our temperate rainforest. So we can see where we've got rainforest with very, very high annual precipitation and very low evapotranspiration, but for cool temperatures. And that corresponds to what is referred to as a temperate rainforest, as opposed to tropical rainforest. So we can recognize temperate rainforest as an ecosystem or a biome type in these kinds of schemes. So another way of defining temperate rainforest climatically, an important paper that attempted to do this was published in the early 1990s by Paul Allerbach, and he suggested that temperate rainforest occurs where there's over 1,400 millimeters of precipitation a year, where the climate is relatively cool. So the mean temperature of the warmest month, July in this case, is less than 16 degrees, and it's consistently moist. So over 10% of annual precipitation occurs during the driest part of the year. So this is a very widely applied definition of the conditions under which temperate rainforest can form. So we can take a definition like this, and we can ask where do these conditions occur in the UK? Do these kinds of very wet conditions occur in Scotland, and whereabouts? So when we do that, this map here with the splodges of green, this shows where temperate rainforest occurs according to this definition. So we can see large areas of temperate rainforest along the west part of Britain. And this other more colourful map is a map that divides the climate in a slightly different way. So it shows the difference between an oceanic climate, which is very, very wet, but with moderate temperatures, where summer and winter temperatures are very similar, versus a continental climate, where the climate is drier and you have more temperature extremes. And the areas in green and blue show where we have an oceanic climate. And this area of deep blue in Western Scotland shows Britain's most oceanic, hyper-oceanic climate, which represents the climatic zenith for the development of temperate rainforest in this zone in Western Scotland. So we can take these schemes and we can then plot them globally. So we can say, well, where does temperate rainforest occur across the globe? And what's important with these different life zones, these different ecosystem types that are defined by the climate, is we've got all these polygons and they all look about the same size on this diagram. But actually, some of these climatic conditions are more common than others. And the set of climatic conditions that allows the development of temperate rainforest is actually quite rare on the planet. And so it covers less than 1% of the global land surface. And that's compared, for example, to tropical rainforest, which covers about 7% and then boreal forest, which covers about 15, 16, 17%. So temperate rainforest, the climatic conditions that are suitable for temperate rainforest, that kind of bioclimatic zone is very rare. And then globally, Europe has about 15% of that climate space, of that potential temperate rainforest resource. And 40% of that occurs in the British Isles. So the British Isles have got the bulk of the suitable climate space for temperate rainforest as it might occur in Europe. So what does this temperate rainforest look like? Well, it's our ash woods and our oak woods on the west coast of Scotland, beautiful woods, often with these short, gnarly trees like these oak trees in this example here, or our hazel woods, open canopies, beautiful ground floras, really charismatic habitats. But I really want to make the point that the difference between a woodland and a temperate rainforest is down to the lichens and bryophytes that occur there, and that particularly occur there as epiphytes. And we find these absolutely stunning epiphytic communities, very rich in lichen diversity in our temperate rainforests. And we can see that quite clearly if we take another look at our global map, our global distribution of temperate rainforest, and we think about the distribution, say, of our oak trees. So Quercus robur here, and we can see that the distribution of Quercus in Europe and Eurasia is very broad. So it's not just within the temperate rainforest zone. The temperate rainforest isn't actually defined by the presence of oak, and it's not defined simply by the presence of hazel, which is widely distributed, more widely distributed than that temperate rainforest climate. But when we look to some of the lichens, so Pseudocyphalaria norvegica, for example, we can see that these lichen species are absolutely restricted to this temperate rainforest zone. And they have these remarkable distributions where we've got species distributed in southwest Norway, western Britain, down through the Azores and into South America. Or here with Crutandini petractoides, we've got another remarkable distribution in southwest Norway, western Britain, and then across to western North America. So absolutely defined, characterized by our lichen and bryophyte epiphytes. And we see these distributions over and over again. So we see that hyperoceanic zone on this map here, and the distribution of Pseudocyphalaria crocata, very restricted to this extremely oceanic zone. And different, therefore, from the tree species, but we find those skewed distributions over and over again for our lichens and bryophytes. Okay, so given that these lichens, and I'm going to focus on lichens now, given that these lichens are so important in making a temperate rainforest what it is, they're the unique elements that we find when we enter into a temperate rainforest. Let's think about, in the second part of the talk, let's think a little bit about their biology. So the important thing about a lichen is that it's more than one thing. So if we, I guess many people, all of you will have seen lichens, and you can pick them up and they look like a little plant or something. If we take one of those lichen thalli, the body of the lichen, and we slice it like we're slicing a carrot, put it under a microscope, then we end up with a cross section that looks a little bit like a sandwich. And in this cross section, we have an upper surface made of fungal material, a protective upper layer called the upper cortex. And then the jam in the middle here, these green blobs, those are algae that are sitting within the middle of the lichen. And then we have a lower section of more typical fungal cells, these stringy hyphal cells in the medulla. And in this case, we have a protective lower cortex also. So the bulk of the lichen, most of the biomass of the body is built out of fungal material. Most of the fungi that make lichens occur within a group called the Ascomycetes, which is a sister group to the mushrooms and toadstools. And the Ascomycetes produce a range of different fruit bodies, but many of them look like little jam tarts. So they have these very cute little jam tart-like structures with a kind of pastry crust edge and a disc in the middle. And in the disc, if you look down a microscope, then you have a flask where the fungal spores are produced. The flask is called an ascus, and that's typical of the Ascomycetes. And the name that we give to the different lichens is actually they're fungal names. So the different lichen species are actually different fungal species. Now the fungi are a little bit like us, the animals, and they need to absorb a source of food from outside their own tissues. And lichens are fungi that have found a way to do that by building these special structures inside of which they can farm a population of algae that are photosynthesizing or a population of cyanobacteria that are photosynthesizing. So these algae or cyanobacteria are photosynthesizing, and then the fungus is protecting that population, and it's harvesting some of the sugars and the carbohydrates away from the algae or the cyanobacteria for its own food. So these could be algae, as I said, they could be single-celled algae, or they could be photosynthetic cyanobacteria. If they're cyanobacteria, then the cyanobacteria often produce these specialist cells called heterocysts, where they're fixing nitrogen from the environment as well. So they're fixing nitrogen directly from the atmosphere and making it available to the fungus. Okay, the other important thing about the lichens that they're what's referred to as a polyhydric, and that means that they don't have a complex vascular system. They don't have xylem and phloem like vascular plants. And when the environment is dry, then they tend to dry out. And when the environment is wet, then they get wet. They're very good at drying out. And when they do dry out, they can survive for long periods of time when they're dry, and then they're dormant. But they require liquid water. They require water, sorry, in order to become metabolically active again. So in this graph here, what we're looking at is time along this horizontal axis, and metabolic activity on this y-axis in terms of carbon dioxide exchange. So what we see here is a green algal lichen that was introduced into a chamber with high levels of water vapor, so high humidity, and just with the availability of water vapor, this green algal lichen can get wet again, and it can start to photosynthesize. So we've got positive carbon dioxide exchange here as it's photosynthesizing, and it can start to respire. So the lichen needs to get wet in order to become metabolically active, but it can do that. The green algal lichen can do that with water vapor. And that's different from the cyanobacterial lichens. So we see the same experiment here, but with a cyanobacterial lichen, a peltigera. And when the lichen is introduced to this humid environment, nothing happens initially until this little arrow here after 24 hours where liquid water is introduced. And it's only with the introduction of liquid water that this cyanobacterial lichen becomes active and starts to photosynthesize and starts to respire. So the green algal lichens can become active again with high humidity, whereas the cyanolichens require liquid water in order to become active again. Now I've said that the lichens, when the environment is dry, then they dry out, and when the environment is wet, then they get wet. That's important, but they can kind of they show adaptations to different moisture environments based on their kind of morphology, their body plan. So they're not totally at the mercy of the environment. There are many different shapes and sizes, and the different shapes that and colors and sizes that they represent can be adaptations to different kind of drying and moisture environments. So the shape and the structure of lichens is often expressed using two metrics. So specific thallus mass, which is the dry mass per unit area. And if you think about it, that's almost like the surface area to volume ratio. So how much surface area is there per unit mass? So how branched or how thin are they? And then the water holding capacity, which is the amount of water that they can hold, again, per unit area. And with these two different metrics, we can characterize the different shapes and sizes of lichens. So if we've got very, very low specific thallus mass and very low water holding capacity down here, these are often the lichens that are very thin and very, very highly branched, like our Bryoria or our Alectoria species. If we move to lichens that have got a slightly higher specific thallus mass, so a little bit more mass per unit surface area, then that might be represented by some of our foliose lichens, like our Parmelia sulcata here. And then we have lichens that have huge water holding capacities that can take up, per unit surface area, they can take up massive amounts of water. And these are often our cyanobacterial lichens that require that liquid water, that lots of water in order to become physiologically active. So things like, in this case, a Peltigera species, they really swell a lot when they soak up lots and lots of water. So we can use these metrics to kind of characterize different lichen body plans that represent different adaptations to the environment. And in a famous paper by a Norwegian scientist, Ingvar Gausler, he suggested that water is available to lichens in three different ways: as rain, as dew, or as humid air, and that we can characterize the adaptations of lichens with respect to their use of these different moisture regimes. So if we've got rain here and we've got dew and we've got humid air, then he suggested that the cyanobacterial lichens will occupy environments in which we've got lots of rain, lots of liquid water or heavy dewfall, whereas the green algal lichens will be more dominant in areas where there might be less dewfall or where it's quite dry and they have to rely on water vapor. And then likewise, with respect to the morphology and the body plan, these lichens that can soak up lots and lots of water and really swell full of liquid water, they will characterize environments where there's a lot of rain or heavy dewfall. And then these thin, very lichens with a very high surface area to volume ratio will occur in places where they can access, where there's less liquid water available and where they rely on water vapor. And then if we think about our temperate rainforest, what we're going to find is that with these very, very high levels of liquid water, rain and fog, the kind of lichens that we find there will be adapted into this kind of area here. Lots and lots of liquid water. So cyanobacterial lichens that can soak up lots of water. And indeed, these cyanobacterial lichens are really, really important. They're a really important part of the epiphyte biomass and the epiphyte diversity in our temperate rainforests. Final thing I want to touch upon with respect to lichen biology is the reproduction and the dispersal. So we've already mentioned that these are often ascomycetes that are producing spores. There's an example of some spores, and there's the little fruit bodies, and they're producing the spores in these flasks. The spores are microscopic and they're dispersed into the environment. Being small, they can disperse a very long way. So you've got the advantage of long distance dispersal, but then wherever they end up, they have to resynthesize with a compatible alga or a compatible cyanobacteria to start to form a new lichen. The other way that lichens reproduce is by using structures that effectively bud off the lichen, a sort of clonal reproduction. And these are referred to, they're either like little stalks called isidia or powdery granules called ceridia. And the key thing about these is they include both the fungus and the algae or the cyanobacteria. So they tend to be larger, so they may not disperse as far, but wherever they end up, then you've got the algae and the cyanobacteria and the fungus. So you've got that compatible symbiosis that can start to form a new lichen straight away. Okay, so we've talked about temperate rainforest and its rarity and its importance globally, its biogeography. And we've introduced ourselves to lichens that really characterize that temperate rainforest. Such an important part of its biodiversity, really what makes a temperate rainforest. So we're going to take that information now, and I want to think about just three pieces of work that help us to understand how we might protect or lessons about how to protect this diversity. And this is all about resilience. It's all about the resilience of these rainforests given future environmental change. So the first really important point is that we have to protect what we've already got. So we have to work really hard to protect the remnants of temperate rainforest that still survive. So this is a map from the Millennium Ecosystem Assessment, and the areas in red show areas that have been deforested. So areas that would have had forest and the forest has been cut down. And we can see that deforestation is just, it's extensive across Europe, including within Britain. And then this shows a little piece of work that we did where we looked at the extent of native woodland in the landscape across a longitudinal gradient from eastern Scotland to western Scotland. And we can see that the extent of native woodland goes down as we move from eastern Scotland to western Scotland. So we have less native woodland in western Scotland in that temperate rainforest zone. And there's also a relationship between the extent of native woodland and the size of individual woodland patches. So not only do we have less native woodland in western Scotland, but the native woodland that we have is smaller and more fragmentary. And an estimate by the Alliance for Scotland's Rainforest suggests that we only have 30,000 hectares of these temperate rainforest remnants left within Scotland. So these are small and isolated fragments of woodland that are left. Now, what we were interested in is the effect of that isolation on species the rainforest species that occur in these woodland patches. And the nice thing about this is we're able to use record data. So the kind of data that people go out, they collect species lists, inventories for particular places, they submit those records to the BSBI, the BSS, the British Lichen Society or the British Biological Society, and you can accumulate records over time. The challenge with analysing these records is that they're often patchy, and the sampling effort can change over time. But there are some methods that were developed by colleagues at the Centre for Ecology and Hydrology that help us to extract trends from the kind of records that botanists and lichenologists and bryologists provide. So the challenge is that we're interested in records for some of these rare species that only occur at a small number of sites in Western Scotland. And the recording effort for these species is very low. But what we can do is we can use much more common species and the records for much more common species that are kind of ubiquitous in the landscape as a measure of sampling effort. So if people are going out and they're recording epiphytes, there's a whole bunch of species that kind of occur everywhere. And if people are going out and recording epiphytes, then they will inevitably tends to record those species. And so you can use those species to look at changes in recording effort over time. And then you can ask how the number of records for the species we're interested in changes with respect to that recording effort. So recording effort for the common species could go up. And it might be that the number of records for our target species that we're interested in goes up, but not as fast. It could be that recording effort for the common species stays the same. And it might be that recording effort for the species that we're interested in goes down. It could be that recording effort for the common species goes down over time. And then it could be that the number of records for the rare species, the threatened species that we're interested in, goes down at a faster rate. And all of those would represent what's referred to as a negative change index. They would represent a situation in which the number of records for the species we're interested in is falling with respect to the recording effort. And when we look at a range of species, these are particularly species associated with woodlands of long ecological continuity in this green zone on the west of Scotland. When we do this type of work for these temperate rainforest species, we find that consistently from decade to decade over the last 50 years, we have a negative change index, which would suggest that these temperate rainforest species in these small and isolated pockets are continuing to decline over time. So they're not protected. We still need to work hard to protect what we have left. So as well as protecting what already exists, and that might be remedial action to, take care of individual populations for these very rare and threatened lichens. We also inevitably want to increase the temperate rainforest resource from the position we're in, where it's dwindled down to these small and isolated patches. And we've got a massive opportunity to do this now with things like Scotland's forestry strategy, which has an aim to increase the amount of forest and woodland cover over Scotland through to the 2030s. And that includes 3,000 to 5,000 hectares of new native woodland per year, and also increasing the amount of woodland that's in good condition. So it's not just about planting new woodland. A woodland like this, which is choked with rhododendron, is effectively invisible to a lot of the important lichens and bryophytes. And actually, by removing rhododendron and putting our woodlands back into good condition, we're basically making new habitats available again. But there's a tremendous opportunity with policy to increase the amount of temperate rainforest that we have in Scotland. And so my two final examples are around that expansion. And this example suggests that what we need to do when we do this is expand around existing remnants of rainforest. So what we did here was we were sampling the air to understand how these species are distributed in the landscape. So you can see these little devices that are positioned here. These are air samplers. They have a set of arms that spin round and round, and they've got sticky stuff on the arms. And as they spin, they're capturing the spores and the isidia, the ceridia, the propagules that are flying around in the air. And then we can scrape the sticky stuff off and all the stuff it's captured into a tube, and we can use the DNA to identify individual species. Now, what we can do with that is, if we've got a population of a species we're interested in, and we put an array of these samplers at different distances away from that population, then we capture the propagules and we do the DNA sequencing to understand how many propagules we're capturing, at what rate the propagules are traveling through the landscape. Then we can start to understand that dispersal, and we can think about how near or close new woodland would have to be in order to be colonized by some of these rare rainforest species. And when we do that, this shows the results of that kind of analysis. So on the x-axis here, we've got time. in years, 20 years, 40 years, 60 years, 100 years, 200 years. And then on the y-axis, we've got the number of trees that are colonized in a new woodland. So if we're planting a new woodland, how many trees are colonized by our epiphyte species over time? And we can see that indeed over time, the new trees and the new woodlands start to get colonized. But there's a big difference between woodlands that are planted closer than 100 meters and woodlands that are planted further than 100 meters away from a source population. So if you plant further than 100 meters away, colonization into that woodland is more limited. And if it's less than 100 meters, then colonization into the new woodland is more rapid. So this kind of work tells us that the best way to expand rainforest, if we're interested in the recolonization of some of these rare lichen epiphytes, is to expand around existing populations, so they have the opportunity to expand and for their populations to get larger. And then my final piece of work is with respect to climate change and resilience to climate change. So we're talking about an ecosystem, temperate rainforest, that by definition is restricted to a very particular set of climatic conditions, and that should immediately raise concern about the effects of climate change on this kind of ecosystem, this kind of habitat. So the Earth is warming, of course. We've got warming temperature trends, and then in Scotland, if we look at the projections for Scotland, and this is with respect to summer precipitation and summer temperature here, then what we find is that summer precipitation, the amount of rainfall in the summer, is expected to go down, while temperatures are expected to go up. So increased periods of drying in future summers as a consequence of climate change. And remember our cyanobacterial lichens and how dependent they are on liquid water, falling into this zone where they're adapted through their ability to take up water, through that necessity of being associated with cyanobacteria that need that liquid water for photosynthesis. Our rainforests are characterized by species that rely on a steady supply of liquid water. So those kind of drying periods, future drying periods in the summer, represent a potential threat to the cyanolichens that really occur in our, that are an important part of our temperate rainforest. So we looked at the way in which we might exploit the landscape in order to offset that drying effect and the negative effect of drying on cyanolichens. So to begin with, we looked at the consequences of the drying for cyanobacterial lichens in Western Scotland. And I'm going to show you a series of maps that show the effect of climate change over time. And areas in blue show areas where climatic conditions remain more or less suitable for cyanobacterial lichens, for these cyanolichens. And as the pattern shifts to green and orange and red, then we start to see a negative effect on these species. So we can see lots of blue through to the 2040s even, habitat suitability, climatic suitability for these cyanolichens. But as we move through to the 2050s, climate change starts to bite, 2060s, the 2070s, and the 2080s, we see increasing negative effects in these models of drying climate on cyanobacterial lichens. But what we also found is that we can offset that effect by planting new woodland in places that maintain high levels of water and high levels of humidity. So this shows the amount of new woodland, the proportional increase in new woodland that we would need to plant to maintain cyanolichens with their current status, in the landscape. And we see that we need to plant a lot of new woodland, but we plant that woodland, if we plant that woodland around rivers and streams, so within riparian zones, then that fits within current planting schemes, current planting targets. This graph here shows the amount of additional woodland we would need to plant beyond our current planting targets as we increasingly expand our planting away from riparian zones, from rivers and streams. So we can see that by targeting the planting to areas that maintain constant moisture levels, we effectively gain more for an equivalent amount of woodland. So we maximize the benefit of new woodland, considering the future effects of climate change. And this is an area of real concern for us at the moment. We're doing quite a lot of work on this, trying to understand how we might position woodland in landscapes in order to ensure that these future woodlands have the right kind of microclimates within them, so that this rainforest diversity can continue to survive and exist in our temperate rainforest, even as the future large-scale climate continues to dry. And we might do that by thinking about positioning woodland in a landscape. It could be with respect to altitude, it could be aspect, it could be distance from a water course, but within any given landscape, there's a whole range of microclimates that we can exploit in the development of new woodland in order to create these microclimates. And then the structure of the woodland internally, we can think about aspects, tree lean, canopy cover. Again, we can work with the structure of the woodland to maximize the benefit for our temperate rainforest species. Okay, so to conclude, we introduced Scotland's temperate rainforest. It's globally important, it's globally rare, and we have strong representation of it within the European context, definitely. It's characterized by its lichen diversity. I've spoken mostly about lichens, but a lot of what I've said is transferable to bryophytes. I apologize to the bryologists, but it's characterized by its lichen and bryophyte diversity. The key things are to protect what remains. That's unfinished business. The lichens and bryophytes in our existing temperate rainforest are still threatened, and we need to work hard to protect those populations so they represent a source for dispersal into future new woodland. And when we expand that woodland resource, we might do that by expanding around existing high diversity remnants to make sure that we get colonization into that new woodland. And we need to do that in a way that considers microclimatic variability and the availability of future habitats under climate change. And with that, I want to finish by thanking the co-authors and partners who were directly involved in some of the work that I've presented. So that's Becky Yar, Sally Eaton, Brian Coppins, and Dan Hayden from the University of Glasgow. And if you want to find out more about rainforests, Scotland's rainforest in more general terms, then have a look at the website for the Alliance for Scotland's Rainforest. And there's a lot of information there about all sorts of different aspects of okay, thank you very much.
That was wonderful. Thank you very much. That was very interesting. I learned an awful lot there. We have some questions. Do the fungi ever expel the algae or the cyanobacteria when stressed, just the way coral and plankton do?
Gosh, that's a really, really good question. So, no, my understanding of this at the moment is that that's not the case. There are very few experiments on this though, and one of them was done by a student who did an MSc with us actually at the Botanic Garden, but then went to do a PhD in Germany with Claudia Colesi, who now works at the University of Edinburgh. And they did some transplant work where they transplanted lichens into different climates, and then looked to see whether they switched their photobionts. And that didn't seem to be, if I recall correctly, that didn't seem to be the case, that there wasn't this kind of switching of photobionts. So they kick one type out and then they bring another type in. But that does appear to happen over, over generations. So colonization, if fungal spores are colonizing into a new habitat, or they're colonizing into the same habitat, but over time, then that's the opportunity when selectivity can happen for a different kind of photobiont. So, so they don't, as far as we know, they don't kick them out and then build them back in again. Someone will correct me about this, but they do it. They do it generationally. So you do see patterns where the same lichen fungus will use different photobionts in different parts of its range, and those photobionts are optimally adapted to the different environments. So they effectively kind of allow it to have a wider niche than it might otherwise have.
Good, thank you. There's another question here about photobionts. If spores land and fail to capture a suitable photobiont, do they simply die or develop into normal fungi? I mean, I had the same. I thought, if when you were looking at the dispersal distance, do the fungi and the bacteria and the algae all disperse the same distances, or do they pick up new variants or new types of the same species in the new place? I mean, you partly answered that just now, but will the fungi just die, or will they develop a new photobiont?
Yeah. So, so yeah, this is another good question. But both of these questions are still like the topic of active research. So these are kind of like, you know, proper research questions. So the lichens didn't evolve once. So they're not this. So it's this being a lichen is a fungal strategy. It's a way that fungi access food, and it's evolved multiple times across the Ascomycete tree of life, and you get Basidiomycete lichens as well. So so it's evolved multiple times, and also you get different levels of being a lichen. Now, most of the lichens that we talk about build these complex structures with these particular body plans and morphologies, and they are tied into that lichen lifestyle. And if their spores land somewhere, they often have quite broad tastes in the kinds of photobionts that they might associate with. So quite common algae. But if they do land somewhere, they can grow for a little while by themselves. But ultimately, if they don't find the right photobiont, then they will die. However, there's a set of fungi around the edges of being a lichen that can adopt a very, very simple relationship with algae or cyanobacteria, and they'll make a very simple lichen that's not as complex as the ones we've been looking at in this talk. And some of those can make that association with the algae, but can also have an alternative source of food. So they might be saprophytes, for example, or they could be parasites. So yeah, so there's a whole sort of suite of lichens. Some of them are very specialist, they'll only ever be lichens, and some of them around the margins of that evolutionary transition can be more than one thing.
Wow, it's far too complicated for an hour's talk. We need to have more talks on specific aspects, I think. So Lynn Jones asks, is there a correlation between hydric requirement and pollution tolerance of different lichens?
Oh my gosh, yeah, another good question. Yeah, I mean, there's yes, and so, yeah, it's more complicated than that. So the ones that have got a very high surface area to volume ratio, like the very branchy ones, like the Briorias and the Alectorias, they tend to be quite highly sensitive to pollution, and I guess that's because they're they've got that high surface area for the absorption of atmospheric pollutants. The species associated with cyanobacteria, however, also seem to be very sensitive to pollution. And then there was a study where they looked at the hydrophobicity of the lichen surface, so how water repellent it is, or how easily it absorbs water. And the ones that were more hydrophobic were more tolerant to pollution. So there is a relationship, but it expresses itself in different ways. It's not just a kind of straightforward correlation.
There's more research needed.
Yeah, more research.
Answer to every question. Nathan asked, is there a relationship between the propagule type, the spores or the insidia, the soredia, and the rates of dispersal between patches?
Yeah, there seems to be a relationship when people look at that at ecological scales. So if we're thinking about woodland patches and woodland ages within a landscape, then smaller propagules disperse further and more easily colonize into new habitats. It appears to be a bit more complicated though when we look at longer time scales. And actually some of the some species that are asexual and they reproduce with Soredia or Isidia often have very, very broad biogeographic distributions, which is a bit curious. And so it kind of bucks the trend a little bit. It's quite interesting. I'm not sure we know why exactly, but it might be that they have the advantage of taking with them a compatible photobiont, so that when you get these very rare long distance dispersal events, they're well positioned to start growing. But then there's also an open question about the identity of some of these things and whether they're really the same thing or whether they're actually different species at that kind of biogeographic scale.
Yes, I said, well, a related question from Maria Chamberlain. Since the lichens, or at least the fungal partners, can reproduce both sexually and asexually, does the environment act as a switch hitter, affecting the balance of resources committed to these different strategies? There's so many questions arriving here now that they keep disappearing off the off the screen.
Yeah. So often lichens will either reproduce sexually or they'll reproduce asexually. So a lot of species will only do asexual reproduction. A lot of species will only do sexual reproduction, but some species do both. And there's a really brilliant paper in I think it's in Oikos from the early 1990s. I can't remember the author, but they looked at they took a lichen that reproduces asexually and sexually, and they observed its population growing on a slab of rock, and it colonized the rock with a few thalli, and it was reproducing asexually, and those asexual propagules spilled out onto this rock surface, and it started to fill it in. And as it filled in and the lichen thalli started to touch each other, it switched to sexual reproduction, which they interpreted as like an escape strategy. So the local habitat was kind of full, and then there's some kind of switch that allowed sexual reproduction to happen, which is just
it's fascinating.
It's almost like too good to be true.
Yes,
I've never seen it repeated, but it's a great study. Yeah.
Yes, no, that's great. Do they disperse the insidia at a particular time, or is it temperature and humidity or environmentally determined, or is it seasonally? Or what?
So I'm not sure about the Isidia and the Soredia, but there are studies that have looked at the production of spores in the fruit bodies, And there is some evidence that there's a phenology to that, and that the kind of wetter times of the year, autumn and spring, are the times when they're most active in spore production. Yeah.
So somebody, I'm not quite sure how to pronounce the name, Hebe Karas, said was really interested in your graph showing the huge step change in lichen colonization of new woodland more or less than 100 meter away. And you also explained that for climate change resilience, planting woodlands should be targeted in riparian zones. So obviously the ideal scenario was new woodlands and wet microclimates within 100 meters. But is there a way to solve where this is not possible? Would seeding of the lichens into new woodland more than 100 meter from established lichen-rich woodland work?
Yeah, so the 100 meters is because of the lichens that we were targeting. So we were purposefully targeting lichens that we know are rainforest specialists, but we know that they're rare and very isolated in the landscape. So we were particularly looking at lichens that have that dispersal problem, and what do we do about these species? Because these are the ones that are the most threatened and the ones that really need, they really need the advantage of new woodland. So we were looking at dispersal limited lichens. So that's not true of all, that's not necessarily true of all lichens, but it is true of, some of these rare and threatened species. And we're also doing studies where we're attempting to translocate these species to understand whether we can move them into new woodlands purposefully. And the results from that look promising. So we think we're able to do that. I don't want to get too, maybe I should clarify with the riparian zones. We use the riparian zones as an example because it's an obvious kind of place where if the global climate is drying, it's an obvious kind of place where those high levels of moisture might still exist. It's an obvious landscape feature, but there are other landscape features. So we looked at, for example, the aspect and the angle of slope. And you, by planting, by reforesting into certain slope and aspect situations, you can also create those kinds of benefits. But then, like I said, the structure of the woodland can be really important too. Mm-hmm. It's not just riparian zones, but I think this is still, I think trying to understand how we can manage woodlands in order to create and protect those microclimates into the future it's still a work in progress.
Yes, it's very complicated. I mean, one of the issues is if you focus on riparian zones and then climate change causes the river to dry up, then it might not be the best place to have put them. And
yeah, I mean, that's
higher altitude where it might be wetter longer.
Yeah, we've not even considered that. Mm-hmm. I mean, there's probably a broader collaboration with geomorphologists and hydrologists where we could do something like that. Yeah.
Mm-hmm. So Alex Twyford said, "What a wonderful talk, Chris." Oh, and then it disappeared. How resilient are temperate rainforest lichens to pollution? It seems like there's still abundant lichen growth near settlements on the northwest coast of Scotland, but is this just biomass and not diversity?
So they're not resilient to pollution. I can only imagine that in northwest Scotland the air is relatively clean, and it's clean enough to permit the lichens to continue to survive. Yeah. I mean, there's been, so it's an interesting question because often where you've got a very high rainfall environment, you can end up with quite high deposition for things like nitrogen, but the actual concentration of nitrogen in the rainfall is quite low. So the concentration seems to be important as well. But they're very sensitive, and nitrogen pollution is a real concern.
So I guess it's cleaner there because of the prevailing wind and
I think that's right.
Distance across the sea.
Yeah, that's right.
Yes. So Chris asks, do we ever see production of anthocyanins in lichens in response to environmental stress?
Gosh, I don't know. I'm sorry. Yeah, I'm not sure.
Yes, because I guess if color is used to identify them.
Yeah. So I'm not anthocyanins. I'm not super familiar with those, but you do get this. You see, I'm not very good at chemistry. The thing, I don't know if anthocyanins are related to anthraquinones, for example, but they do upregulate their metabolites, like their chemistry, in response to certain triggers. So, so sunlight, for example, the xanthoria. I showed a picture of the yellow xanthoria, and you can even see on a twig, if you find that lichen on a twig, you can see that on the top of the twig, where it's exposed to the sun, it will be very, very yellow. And as you move around the twig and it gets more shaded on the underneath, it will get green. People have done experiments where they put them in different environments, light environments, and they upregulate or downregulate the production of those chemicals that act as a UV protectant. So, yeah. So
it might be.
Absolutely. Well, yeah. They definitely, they can definitely turn those chemical pathways. It seems as if they can turn them on and off, but anthocyanins in particular, I'm not sure.
Okay, and one from David Chamberlain: is woodland connectivity an issue for the future conservation of the temperate rainforest?
Yeah, I think so. I think that's right. I think yeah, in the sense that we want to increase the population, in the sense that we want to increase the population sizes, and we want to ensure connectivity between populations so that we have these viable, well, larger populations are more resilient. And then if they're interconnected with gene flow, then we have viable metapopulations. So I think that's super important from the population point of view, but not in the sense that they're going to migrate somewhere. So we've done some work recently where we looked at the climate in that temperate rainforest zone, and we looked at projected future climate conditions across Europe to see where there might be a sort of future analog for the temperate rainforest today. And there isn't one, as far as we can tell. So the climate is going to change, but it remains its own closest analog. So there isn't somewhere where these species can migrate to somewhere else in Scotland or Europe.
So in the future, might there be a place for them? I mean, because the distribution of the rainfall and drought is going to change. Places might become wetter, longer, potentially.
Yeah, I mean, I picked on the cyanolichens in this example and the summer dryness, but there are other kinds of species in the rainforest, and some of them are species that say they're associated with a particular kind of alga, Trentipolia. There's very, very high diversity of Trentipolioid lichens in the subtropics and the tropics. And then we have species that occur in genera, where these genera are much more common, again, in the tropics and the subtropics. And these are species that are probably sneaking up into Western Britain because of the mild conditions. And so the element of warmth for some of these species, future warmth, might actually be an advantage, but only if it remains wet and moist. Yes. So it's that interplay of temperature and moisture that is really tricky.
Yes, I was thinking if they're not well connected and there's more fragmented, you have drier edges, more edges effects than you would have if they were more connected into a bigger patch. So that might be another issue of connectivity.
Yeah, and actually thinking about that, I think I might revise my answer to David's question. There's a whole bunch of species, in fact, that, and this is probably true of the bryophytes as well, that you find down in the southwest of England in a sort of warmer temperate rainforest zone. And so there's a question about if we've got this warming climate in Scotland, will they be able to find their way? Yeah, I've changed my mind about that.
Okay. food for thought. And we need to let you go. It's already past 7 o'clock, but there's a last question from Alice Broom. How old do the trees and woodlands need to be before they're colonized by the rainforest specialist lichens?
Yeah, that's another really good question. So there are species that seem to be associated with old trees, and certainly with old structure. So kind of old trees in a gladed setting. And I mean, I think this is massively challenging because I mean, some lichens only grow on these really old trees. Mm-hmm. And I showed climate projections through to the 2080s, which really isn't that far away in the lifetime of the tree. So if we're planting woodlands and we're trying to think about, and we're trying, you know, we're planting woodlands with trees that we might hope will be protecting lichens in 200, 250 years time. It's really challenging to think that far ahead in terms of well, you know, thinking about tree disease or
yes,
or climate change in 250 years time. Yeah.
Yes, I was thinking what's happening to ash dieback and how much is it affecting those forests?
Yeah, it's a disaster. Yeah, really. I mean, so many of these cyanolichens that I've been talking about, they preferentially occur on trees that have a relatively high bark pH. Mm-hmm. Will include things like elm and ash, and so they are disproportionately affected by ash dieback. Yeah.
Mm-hmm. Yeah. But so Anne Gascoigne, just to finish off, said thank you. That was a very interesting talk and a great Q&A. She wasn't expecting it to be so much about lichen, but that is what I'm really interested in. So fab. So I think on behalf of everybody, that was a fabulous talk and wonderful questions. And thanks everybody for participating. When Julia closes the talk, I'm afraid, Chris, the thing we forgot to tell you is that it closes for everybody. So we don't get to chill out and have more question and answer afterwards. So you'll be pleased to know you can go now. So thank you very much. And thank you to Julia for hosting and everybody for participating. And we look forward to seeing you at our next talk.
Thank you.
Bye.